Hello, everyone, and welcome to today's webinar, Immune Profiling of Hospitalized COVID-19 Patients. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckmancoulter.com. Let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Josephine Giles, Cancer Research Institute, Mark Foundation Fellow, University of Pennsylvania. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Josephine, you may now begin your presentation.
Great. Thank you so much for that introduction. So I'm a postdoctoral fellow in John Wherry's laboratory at the University of Pennsylvania. And today I'll be telling you about our work in trying to investigate the immune response to COVID-19 in hospitalized patients. And I'd like to thank the organizers for having me, specifically LabRoots and Beckman Coulter Life Sciences. So SARS-CoV-2 is a positive-stranded RNA beta coronavirus. It is the causative agent for the disease that we now know is COVID-19. As of Friday in the United States, there were 9 million cases total. This led to a total of 229,000 deaths. Worldwide, there have been about 45 million cases, leading to 1.18 million deaths. This disease can have a wide range of symptoms, including some of the ones listed here. But what we now appreciate is that individuals infected with SARS-CoV-2 can lay anywhere on this spectrum, from having absolutely no symptoms at all to very severe symptoms culminating in death. And we wanted to understand what is the immune response to this viral infection. Now, we know that effective immunity lies here in the middle. So if you have a robust on-target immune response, that can lead to viral control and eventual tissue repair. On the right-hand side, too much of an immune response can lead to immunopathology. And on the other side of this, too little of an immune response, and by that we mean too low in magnitude or too slow, maybe off target, can lead to viral dissemination and then direct cytopathic effects of the virus itself. And for COVID-19, we don't know where we are on this spectrum. And it's entirely possible that different patients sort of have the needle at slightly different places on this scale. And so that may be one reason that we have some patients with no symptoms at all, and then some patients that require hospitalization. So all of the work I'll be sharing with you today has really been the result of a huge effort across many different labs and departments at the University of Pennsylvania, some of which are listed here. This work was really initiated and led on the clinical side by Dr. Nuala Meyer. So today I'll be telling you about 3 different cohorts. Of course, the first is our COVID-19 patients. These were all hospitalized at the University of Pennsylvania, many of which were in the ICU, but some of which were also on the regular hospital floor. We also have a group of recovered donors. These are donors that had a confirmed SARS-CoV-2 infection, but had not had any symptoms for about 2 weeks. And the third is a group of healthy controls. So we received peripheral blood from these donors that was processed at various locations at the university. The samples were then subjected to our deep immune profiling pipeline. This included high-dimensional flow cytometry, serology, and plasma cytokine analysis. In addition, there were many different parts of the clinical data that we included in our analysis. So when we started to think about how to analyze this patient cohort, as I mentioned, we knew we had about 50 parameters of clinical features. And this included demographic information, as well as medical history and underlying pre-existing health conditions, various laboratory tests, as well as the treatments that the patients were receiving, and various measures of disease severity. So the first thing we wanted to understand was how all of these clinical features related to one another. And in doing this sort of analysis, it would give us a global overview of this COVID-19 cohort. So our analytic approach here was to use Spearman correlation with the appropriate non-parametric statistical test. And the exact test that we used depended on the type of features that were being compared. This is the result of that analysis. And what you can appreciate immediately is there's this large block of correlated features. And within this block, we see several different markers of inflammation, as well as different measures of disease severity. Also included in here are some underlying pre-existing health conditions, such as acute kidney disease. And if you go a few rows down, what you can also see is the coordination of that block with age and mortality. So we can pull these individual features out and look at them. So this is looking at several different serum proteins that are associated with inflammation. And what you can see is most of these COVID-19 patients are above this green healthy donor normal range. So what this analysis showed us was that our COVID-19 patients had a high inflammatory signature that correlated with many measures of disease severity, as well as age, and some of the underlying comorbidities. So now we wanted to ask the question, what is the immune response to SARS-CoV-2 infection in these hospitalized patients? And in this study, we focused on 3 different arms of the adaptive immune system. First are the B cells. These are the cells that are largely responsible for making antibodies that neutralize the virus. CD4 T cells. These are T cells that provide all kinds of critical help to the other cells in the immune system. And CD8 T cells. These cells are responsible for directly killing and eliminating virally infected cells. And we profiled these cells with a high-dimensional flow cytometry panel that was then analyzed in the BD Symphony. And in total, our panel covered 27 different parameters. The first thing we looked at was the relative proportions of these major immune subsets in their circulation. What we found was in our COVID-19 patients, there was an increase in non-lymphocytes and a simultaneous decrease in both B cells and T cells. And when we looked within the T cell compartment, we found that this decrease tended to be skewed towards the CD8 lineage. So we were losing more CD8 T cells than CD4 T cells. This loss of lymphocytes is also reflected in the clinical data. This is looking at the all lymphocyte count. And what you can see is approximately half of our patients are clinically lymphopenic. So we wanted to understand, of these remaining lymphocytes, what is their phenotype? We can analyze this flow cytometry data in a couple different ways. The first way is basically using manual gating based on a priori knowledge. And here we can identify known population subsets, as well as perform functional phenotyping. So we'll start with the CD8 T cells. Here we use 3 different surface proteins, CD45RA, CD27, and CCR7, to identify various subsets of naive, effector, and memory cells. And this is enumerated on the bottom. And what we found was there is a decrease in the naive and effector memory 1 compartment in our COVID-19 patients, and an increase in the EM2 and EMRA subsets. And what this showed us was that there was a general sort of global shift from less differentiated cells to more terminally differentiated subsets. Next, we looked at the activated T cell response. We did this in a couple different ways. On the top, I'm showing you co-expression of HLA-DR and CD38. And on the bottom, proliferation as determined by the expression of Ki67. What we found was that some of our COVID-19 patients had a very robust high CD8 T cell activation and proliferation response. However, there were other patients that had essentially no response that were similar to the healthy donors, and this is enumerated on the right-hand side. And so this result was quite remarkable for two different reasons. One, there is a huge amount of patient-to-patient heterogeneity. So we had some patients with a very large response and some patients with no response at all. And also on the higher end of this activation spectrum, it was really much greater than what we typically see for other human viral infections. So the second method we used was unbiased clustering. And this was a combination of using t-SNE and clustering performed through the Cytobank web tool. And it was really important to us to be able to have an unbiased way of analyzing the data. Because COVID-19 is not very well characterized, we didn't want to necessarily miss anything because we were only looking for the cells that we already knew about. So the first thing we did was to take all the non-naive CD8 T cells that were from the healthy donors, the recovered donors, or the COVID-19 patients, and then we put them on this t-SNE map. So what you can appreciate immediately is that there are some portions of this map where there are cells that are only present from the COVID-19 patients. And alternatively, you can also see areas of this map where you see high density in the healthy donors and the recovered donors that are essentially lost or absent in the COVID-19 patients. To parse some of these differences, we can then use flow-sim clustering on the t-SNE axes. To understand what each of these clusters might be doing biologically and also sort of their cellular identity, we can look at the individual protein expression for these various markers in our flow cytometry panel. But what we really wanted to know is how these various clusters were differentially distributed between our three donor cohorts. So on the top, I'm showing you three different clusters, clusters 1, 2, and 5, that were increased in the COVID-19 patients. And all of these clusters contained EM array-like cells that had differential expression of the transcription factor TBET. Probably the most notable cluster here on the heat map is cluster 14. And this cluster contained the highly activated and proliferating cells. So this was nice because it basically confirmed what we saw with our manual gating. These are the cells that expressed HLA-DR in CD38. But we also saw from this unbiased analysis that they expressed the inhibitory receptor PD-1 and FAS, or CD95. So using this unbiased method, we were able to sort of go beyond what we had already found with our manual gating strategy. And I'll point out one more cluster here, cluster 11. And cluster 11 was interesting because it's the portion of the map that's sort of on the lower right-hand side. It's very prominent in the healthy donors, less in the recovered donors, and then basically totally absent from the COVID-19 patients. And so this cluster had high expression of both CX3CR1 and TBET. So we think these represent basically activated effectors. So now we can apply the same kind of method. Oh, I'm going to skip forward a few slides. So now we can apply this same method to the CD4 T cell response. So first, we're going to look at the activation markers again, HLA-DR and CD38. And so again, what you see is a very robust response in some of the COVID-19 patients. But again, there's almost no response in another subset of the COVID-19 patients that are really almost equivalent to the healthy donors. And you can see that enumerated on the right-hand side here. And I'm going to point out this green line is representing the 90th percentile of the healthy donors. And so all those COVID patients that basically fall on or below that line are not making any response at all. We can then use the same unbiased clustering approach using the Cytobank web tool. And again, we're going to take our non-naive CD4 T cells and create a t-SNE map and then perform flow-sim clustering. And again, we can then look at the individual protein expression of these various clusters. And I'm going to point out two clusters here, cluster 14 and 15 on the bottom. And here, these clusters are increased in proportion in our COVID-19 patients. Cluster 14 is very similar to that activated proliferating subset. They have high expression of HLA-DR, CD38, and Ki67. And just as we saw with our CD8 T cells, they are also expressing the inhibitory receptor PD-1. And then what was interesting is that we find that this cluster 15 that also we believe represent these activated effectors. They have high expression of CX3CR1 and TBET. Instead of being decreased in our COVID-19 patients, like we saw in the CD8 T cells, here in the CD4 lineage, they're actually increased. So taken together, we can basically summarize that we have a robust CD4 and CD8 T cell response in our COVID-19 cohort, but we did see quite a bit of patient-to-patient heterogeneity in terms of the magnitude of the response. So what about the B cells? So when we looked in the peripheral blood of our COVID-19 patients, we saw this massive plasmablast response. Now, these are cells, these are B cells that were basically activated in secondary lymphoid organs, such as the lymph nodes and the spleens. And then in a normal immune response, you will see sort of a transient increase in the blood, and some of them will transit to the bone marrow and eventually form the long-lived plasma cells. So if we compare this to another acute viral infection, such as influenza, we typically only see about 5 to 15% in the blood at the very peak of infection. But here, we're looking at upwards of 40% of all the circulating B cells, so quite massive. And we don't really see this in many other acute viral infections. The only one that we could sort of find that came close was Ebola. So this massive plasma response in our hospitalized patients made us wonder about what the circulating antibodies levels were. So we found that about 70% of our hospitalized patients made anti-SARS-CoV-2 antibody directed towards the spike protein. So next, we compared the level of this antibody to that plasmablast response. IgM is shown here on the left-hand side, and IgG is shown on the right-hand side. And what was really surprising is that there was absolutely no correlation between the plasmablast response and the levels of circulating antibody. There are probably many different reasons for this. The first most obvious one is we were only looking for antibodies directed towards the spike protein. So these plasmablasts may be specific for another viral protein. These plasmablasts also may be lower affinity, so they may be specific for spike, but just below our level of detection for our serologic assays. And the third possibility is they're not specific for SARS-CoV-2 at all. Some proportion of these plasmablasts may either be nonspecific and just responding to environmental inflammation, or they may be responding to some kind of self-protein. And it's going to take a lot more work, I think, to sort of figure out what these plasmablasts are all responding to. So what about the T cells that then help these B cells in making antibody? So typically during an acute infection, you can see a transient increase in the blood of these helper T cells. And these helper T cells are really doing their work in the secondary lymphoid organs with the B cells. But we can find them in the blood using these two different surface proteins, PD-1 and CXCR5. And now, if you drill down on that circulating TFH subset using two additional markers, CD38 and ICOS, we know from previous work that the activated antigen-specific cells are in this subset. So if we look at the proportion of these cells in our COVID-19 patients and our other cohorts, if you look at the total circulating TFH, there is no difference at all between the three cohorts. But if you look at the activated CTFH, we did have an increase in our COVID-19 patients. And what was interesting is that we also had an increase in our recovered donors compared to the healthy donors. And we think this is because these donors had just recently been infected within 2 weeks, and germinal centers and these circulating TFH can be activated for weeks after an initial infection. So next we wanted to compare directly the plasmablast response we saw in the blood with this activated CTFH. And so what you can see here is there's a statistically significant but weak correlation between the plasmablast response and the activated CTFH in the blood of our COVID-19 patients. So then we wanted to compare these activated CTFH directly to the antibody levels, and that's shown here. And again, what we see is a statistically significant but relatively weak correlation. And what this data in sum tells us is that there is probably some amount of T-cell B-cell interactions going on in the secondary lymphoid organs that is leading to the production of this antibody. So now that we have a handle on the immune response to COVID-19 in these three different major arms of the immune system, the B cells, the CD4 T cells, and the CD8 T cells, we wanted to try to understand how these immune responses related to those clinical features that we discussed at the beginning. I'll be showing you one example here, and this is focused on the CD4 T cells. So here we're taking the proportion of all of these manually gated subsets, the CD4 naive T cells, the central memory, the various effector memory compartments, and then the circulating TFH, and these two subsets of activated T cells, the DR38 positive, and then the proliferating Ki67 positive cells. And then we compared them to all of these different clinical features. And I'm only showing you a subset here. The color of the square is indicating the Spearman correlation rho statistic. The asterisks are nominal p-values. And then because we were doing so many different comparisons, we also calculated an adjusted FDR to correct for this multiple hypothesis correction testing. And so what you can see here, I'll point out a few different things. First, CD4 naive T cells negatively correlate with procalcitonin. And procalcitonin is one of these inflammation markers that we can find in the blood. The second are CD4 T cells that are either proliferating or activated. They are both positively correlated with ferritin, again, one of these inflammatory markers in the blood. The activated subset, but not the proliferating subset, was correlated with APACHE3, which is basically a measure of multi-system organ dysfunction. And then the proliferating subset also negatively correlated with the total lymphocyte count. And there were other potentially interesting relationships that didn't quite reach our adjusted FDR cutoff. So sort of all together, we found correlations with CD4 T cell activation and then different elements of inflammation as indicated by the clinical data. So our next question was, how do these immune responses change over time in our hospitalized COVID-19 patients? And so for patients that were still admitted to the hospital 7 days later, we received a second blood sample, and then we could compare the two different time points. So the first part of this analysis was again to use Cytobank to first create two different TISNE maps. The first map included cells that were present in the day 0 samples. And then the second map was cells present in the day 7 samples. And sort of the take-home message here is that you don't see any sort of, dramatically global redistribution of the cellular density on these plots between these two different time points. So relatively few changes over time at this global level. But what about individual patients? So the example I'm showing here is of those circulating plasmablasts. And if you look at the two time points from day 0 to day 7, there's no statistical difference. But if you kind of squint, you can see some lines that are going up, some that are going down, and then some that appear to be staying the same. And we can actually break this out into 3 different groups. So in the top, we see 5 different donors that have both time points that are actually increasing their plasmablast response from day 0 to day 7. The middle group here are patients that remained relatively stable over that 7-day time period. And on the bottom are donors that decrease their plasmablast response from the first draw to the second blood draw. We saw similar sorts of patterns in the T cell responses. On the left-hand side is the activated CD4 T cells, these DR38 positives, and on the right-hand side are the activated CD8 T cells. And again, on the top row, we can see some patients whose response is going up, in the middle, patients whose response is staying stable, and on the bottom, patients whose T cell responses are decreasing over time. And what was sort of remarkable about this result, both in the T cell compartment, but also thinking about the plasmablasts, is that most of these patients that were stable or increasing in their response already had elevated levels compared to the healthy donors. So that meant that these COVID-19 patients not only had elevated B and T cell responses, but they were actually being maintained over 7 days. And that is relatively unusual for an acute viral infection, where we typically see more transient responses visible in the blood. So one thing that I've mentioned several times during this presentation so far is this huge amount of heterogeneity we see from patient to patient. And again, looking at our green line, that's indicative of the 90th percentile for these healthy donors, we have some donors that, some patients that appear not to be making any response at all. So these observations led us to the hypothesis that there might be these sort of global immune phenotypes within our COVID-19 patients. And a related question was, how does this immunologic diversity relate to various clinical manifestations and specifically disease severity? So Dr. Nula and her team had scored these individual patients based on the NIH Ordinal Scale. Score 8 was the least severe, and these individuals had no limitations in their activities at all. But COVID score 1 meant that the patients had very severe disease and had succumbed to the disease. So in order to answer some of these global questions, we needed a way to take all of those hundreds of various manually gated immune populations and integrate and analyze them at the same time. And so the method we chose to accomplish that was called uniform manifold approximation and projection. Most of you probably know this as UMAP. So here we are able to take our approximately 200 immune features from those individual manual gating strategies and put them into the one singular two-dimensional space. And so you can see on the upper right-hand corner are healthy donors and our recovered donors, which are segregated quite distinctly away from almost all of the COVID patients. And if you notice, the COVID patients now are colored by disease score. And you can start to see that perhaps maybe the less severe diseased patients are closer to the healthy donors, and then that the donors on the far end are darker colored and may have more severe disease. But the data in this format, it's sort of hard to ask that question directly. So we took that diagonal data and we rotated it and centered it to these two axes, component 1 and component 2. So now we can directly ask the question, does disease severity relate to component 1? And that's what's shown here. So we can now take each individual patient's component 1 score and plot it all together in this bar plot. So you can see that the healthy donors have, on average, the lowest component 1 score. Then you have the recovered donors, and then the COVID patients in stepwise progression of disease severity. And so we basically had taken these 200 immune features, created this two-dimensional distillation, and created a global immune landscape that we can now use to understand various parts of the clinical features, including disease severity. So now let's compare component 1 and component 2 to the other features in our clinical data. So again, we're using the strategy of Spearman correlation paired with the appropriate statistical test. So what you can see on the top is that component 1 is significantly associated with the NIH severity score both at day 0 and day 7. You can see that it is also significantly correlated with Apache 3, which is this measure of multi-organ system dysfunction. And there's some other interesting sort of nominal correlations here. Some measures of inflammation such as ferritin and IL-6, as well as some of the clinical counts, including PMN, which are the neutrophils, and the whole blood cell count. So now we can take this, total global immunologic map and project back onto it individual immune features. So on the left-hand side are the HLA-DR CD38 CD4 T cells. So these are the activated CD4 T cells. And then we can ask, are these frequencies statistically correlated with component 1 or component 2? And in this case, these features are actually correlated with both, which is why you sort of see this diagonal pattern between the red and the blue. In the middle, we are looking at the TBET positive non-naive CD8 T cells. And this population is positively correlated with component 2, but not component 1, which is why you see this sort of horizontal pattern. And the last example is on the the right-hand side, the plasmablasts. And here, the percent of the plasmablasts in the blood are positively correlated with component 1, but not component 2. So you can sort of go through all these individual features and start to understand the global distribution in this immunologic space. We also wanted to understand how our unbiased flowsum clusters that were determined by Cytobank could then help us understand these two different components. So I'll point out a couple different examples here. Here, cluster 14 in both the CD8 T cells and the CD4 T cells were positively correlated with component 1. Non-naive CD8 cluster, cluster 11, that express both TBET and CX3CR1. So these are the activated effectors. Here, cluster 11 is negatively correlated with component 1, but positively correlated with component 2. And just one more. Oh, nope, that's it. So now, basically, we've taken these 200 immune features, distilled it into a two-axis sort of two-dimensional space. And we can see that while our patients really exist on this full spectrum in this immunologic space, these two components have allowed us to sort of crystallize what we think are two different types of immune response in the COVID-19 patients. So component 1 relates to this immunotype 1. And this is where we have highly activated CD4 and CD8 T cells. We have lower circulating TFH, but high plasmablasts. And this is correlated with disease severity. Component 2, you see correlated with these activated effectors, both CD4s and CD8, proliferating memory B cells. And this sort of immunotype is not associated with disease severity. So I've just described basically two flavors of immune response, but we do know that there are patients for which we don't really see any response at all. So we wanted to understand where they're lying on this map. And so just as a reminder, this green line is the 90th percentile of the healthy donors. And so we have these patients that are lying on or below this line in many of these features that we looked at to try to measure the immune response. So we manually defined these as immunotype 3 and then asked where these fell on this total immunologic map. And so they're colored here in black. And you can see that our immunotype 3 patients are essentially lower for both component 1 and component 2 within the COVID-19 area of the map. So then we asked, if you have this immunotype 3 categorical designation, how does that relate to these various measures of disease severity? And that's listed here on the right-hand side. So our immune type 3 patients basically were negatively correlated with all measures of that NIH ordinal scale, as well as I just mentioned, the component 1, the component 2. So that basically meant that these patients that really weren't mounting any kind of immune response, but were still being hospitalized, actually had less severe disease than some of the other adults in this cohort. And I'm going to point out one other thing here, which is the way that the shapes are represented on this map. So the individuals that are represented by triangles means that those individual patients actually passed away. And so we do have a couple black triangles, which means that we did have some of these immune type 3 patients that did succumb to their disease. But what's important about that is we don't actually know why the patients are dying. And so some patients may be exhibiting extreme pathology due to the immune system, but there are likely other causes that this disease can be fatal. And that's what's sort of represented here by the fact that we have these immune type 3 patients that can still pass away. So I'll give a brief summary for this part of the talk. So one of the most notable things that we discovered in our data was the great amount of immune heterogeneity in COVID-19. Specifically, we saw this massive plasma blast response that we really only saw in other acute infections such as Ebola and dengue. We saw surprisingly that this high T cell and B cell response was relatively stable over time in some patients. And using high-dimensional analysis, we were able to define basically 3 different flavors of the immune system that link to different forms of clinical manifestation. And I think this work has some really important implications and leads to many, many outstanding questions. First, I think the most important thing to realize is that we need to consider the patient's immune response in thinking about how to treat each individual patient. There are now hundreds of trials, probably thousands worldwide, trying different kinds of strategies to treat COVID-19 patients. And you have the whole spectrum from immunosuppressants to immune activating drugs. And what I've shown you here is that some patients don't make any response at all. So those are definitely not patients that you would want to put on immunosuppressants. So we need to be able to come up with a way to quickly assess the immune status of these patients and treat them appropriately. A second major question is, a lot of these hospitalized COVID-19 patients, they are making neutralizing antibody, and yet they're really sick. And so we need to understand why that neutralizing antibody is not able to control the virus in these patients. The third question is what I just brought up, which is why are these patients really so sick? Is it immune pathology? Is it viral mediated? We still don't understand. And it's probably very different for each individual patient. And sort of the last interesting thing that I think about a lot is are these differences related to kinetics? There's some emerging evidence that suggests that early viral control is very important. And part of this is perhaps an interferon response. So it's possible that the patients that end up getting very sick and end up requiring hospitalization are patients that, when infected with SARS-CoV-2, fail to control the virus spread early. And then it basically turns into a runaway train. But understanding the kinetics of this response in different donors is going to be really important. So in the last few minutes, I'm going to tell you a little bit about our new work, which is trying to understand the immune responses to COVID in kids. So at the same time that we are collecting samples for our adult cohort, we are also collecting samples from 2 different pediatric cohorts at the Children's Hospital of Philadelphia. We had one cohort, which is the COVID-19 cohort, and this cohort was divided up into 2 groups, one that experienced ARDS, acute respiratory distress syndrome, and the second that did not. So this helped basically as a measure of severity for COVID-19 disease in this pediatric group. The second group were a group of pediatric patients that were diagnosed with MIS-C. So MIS-C stands for multi-system inflammatory syndrome in children. The CDC definition here are individuals that are less than 21 years of age. They have to present with fever, inflammation, and multi-system organ involvement, and also have a SARS-CoV-2 positive test. But the experience at CHOP has been that these are kids that are, even robustly healthy, absolutely no previous sickness, and then they get really, really sick. And some of the most common features here are fever, rash, GI involvement. And a significant portion of these kids complain of very acute abdominal pain. And occasionally, some of these patients we do know have had family members. They've documented infection 3 to 4 weeks prior to this MIS-C diagnosis. So this is sort of what the caseload looked like in Philadelphia. The adult COVID cases peaked around mid to late April. And then basically 3 to 4 weeks later, we had this increase in MIS-C diagnosis cases at the Children's Hospital. And so just this timing alone sort of raises some interesting questions. First of all, were these MIS-C patients basically infected with SARS-CoV-2 around the same time as the adults, but then have had some kind of long chronic infection? And if that's true, what then is the cause for them to develop these acute symptoms? Is there a secondary event, such as another viral infection, reactivation of an sort of an endogenous chronic viral infection like CMV, or some other kind of secondary event, maybe autoimmune? So just a note about the analysis for this cohort. Our primary statistical comparisons here are MIS-C versus the pediatric COVID-19. But what was really great was because all of these samples were collected, stained, and processed at the same time as the adult, we could then actually use the adult sample as basically a comparator. And this helped us have a framework for understanding these immune responses, both in the pediatric COVID as well as the MIS-C. So just like the adults, we had various features of clinical metadata. So what I'm showing you on the left-hand side are 2 different measures of serum inflammation, so ferritin and D-dimer. And you can see that the MIS-C kids here in the green triangles all have elevated levels of both of these serum proteins, indicating global inflammation. And on the right-hand side, you're looking at the all lymphocyte count. And most of the MIS-C kids are below the healthy donor range and are clinically lymphopenic. We can now use our high-dimensional flow cytometry to study the immune response. And first, we're looking at these measure immune subsets. And what we see in the MIS-C kids is a little bit different from what we saw in both the adult COVID-19 and the pediatric COVID-19, which is an increase in the proportion of B cells. But then similar to the adults, the MIS-C are decreased in the proportion of T cells, both the CD4 T cells, but more dramatically the CD8 T cells. And we can see that the MIS-C kids here basically look like the sickest adults in terms of the percent of CD8 T cells. So now we're going to look at the T cell activation, again using some of those same markers we described earlier. What we can see is that there is a small percent of activated CD4 and CD8 T cells in the pediatric COVID-19 patients. But if you look at the MIS-C examples here, there's a huge expansion of both the activated CD4 and CD8 T cells in circulation. And this is enumerated on the right-hand side. And so if you look down on the bottom, specifically at the CD8 T cells, the MIS-C patients have either on par or even greater levels of CD8 T cell activation compared to the sickest adults. So one of the things that we were interested in was to try to understand if these CD8 T cells were not just activated, but perhaps actually exhibiting signs of T cell exhaustion. And this was, we thought, an intriguing question, especially because of that timing I talked about earlier between when we saw the COVID-19 adult cases come in much earlier in April, and then this MIS-C sort of wave coming in 3 to 4 weeks later, indicating that these kids may have been infected for quite a while. And so T cell exhaustion is a state that can happen in chronic viral infection and cancer when T cells are chronically stimulated and then eventually become dysfunctional. So the first way that we assessed this was to look at the inhibitory receptor PD-1. And so we saw that indeed our MIS-C patients had an increase in PD-1 expression, both in the CD4 T cells shown here on the top and the CD8 T cells shown on the bottom, compared to the pediatric COVID-19. And we know that PD-1 by itself is not a good indication of exhaustion because it can also be transiently increased in activated T cells. So we looked at a secondary inhibitory receptor, CD39. And while it didn't reach statistical significance, our MIS-C patients, the majority of them did have increased CD39 compared to the pediatric cohort. And really, one of the characteristic signs of T cell exhaustion is the high expression of multiple inhibitory receptors. And we know specifically that co-expression of PD-1 and CD39 marks exhausted T cells in adult chronic infection like HIV, as well as cancer. So then we examined the co-expression of both of these inhibitory receptors in the CD8 T cells. And that's what we see here. And indeed, there's a statistical increase in the number of CD8 T cells expressing both of these inhibitory receptors in the MIS-C patients. So the last thing we wanted to understand here was, again, how the immune system was changing over time. And this was particularly interesting in these MIS-C patients because when these kids first show up at the hospital, they're really sick and in a scary kind of way. But luckily, with the right treatments, these individual patients almost always recover and are able to be discharged and do just fine. And so we wanted to understand what the immune system looked like at those different time points when they came in and they were really sick, and also what they looked like when their clinical symptoms were improving. So we had 3 different MIS-C patients where we were able to collect 2 different time points and ask how the immune system was changing over that time. So since the CD8 T cell activation response was sort of one of the most remarkable things about these patients, we first looked at the percent of CD38 and DR positive cells at the first draw compared to the second draw. So that's what you see here. And each row represents one donor. So at the first draw, you see this very elevated CD8 T cell response that is significantly decreased by the second draw. And that's enumerated here on the top plot. And then we looked at several other T cell activation markers, including K67, which denotes these proliferating cells. We looked at PD-1 and also looked at the change in these double positive PD-1, CD39 double positive cells. And basically all of these various measures of T cell activation decreased between those 2 blood time points. We then asked how the clinical features were changing over those time points. And so if you look at the top left-hand side here, that's the all lymphocyte count. So as I noted at the beginning, the MIS-C kids tend to have lower lymphocyte counts and some are lymphopenic. But here what we can see by that second time point, that lymphocyte count has actually recovered to almost healthy normal donor range. We can also look at these various measures of inflammation. And sort of the take-home message here is that while many of the kids do not necessarily reach the healthy donor range, you are starting to see a decrease in this global sort of inflammation that we see. And I'll just summarize this last part of the talk. And what I've shown you here is that this syndrome, MIS-C, is characterized by very high T cell activation, and this really rivals what we see in the sickest COVID-19 adults. I've also shown you some evidence that the CD8 T cells in MIS-C may be exhausted, but it's going to take a lot more work to determine if that's true. And I've also shown you that while there's a resolution of clinical symptoms, this does coincide with the decrease in T cell activation. And of course, that sort of leaves a lot of questions about what the pathology of this disease is and whether this is just sort of coincidental or if this disease is actually mediated in part by this high degree of T cell activation. And I'll just point out, of course, so much more work to be done here with this MIS-C syndrome. And with that, I would like to acknowledge all of my many collaborators. Of course, John Wherry, my PI. And underlined here in the Wherry lab, I've listed all of the co-6 authors on the original science paper. And in green here are my co-authors that have contributed to the work on the MIS-C. This whole COVID blood processing unit on the bottom, and I want to point out Amy Baxter really built this from the ground up in the middle of the pandemic. It was a huge effort, and lots of people contributed, really volunteered their time. Nuala Meyer, who initiated all this work on the clinical side, many other labs at Penn, including at the Children's Hospital of Philadelphia. And I'll just thank my funding sources, especially, of course, CRI and the Mark Foundation. And I'm happy to take any questions.
Thank you, Josephine, for your informative presentation. We would now like to start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started. Our first question is, does hydroxychloroquine alter the immune response in COVID-19 adult patients?
Yep, that's an interesting question. So some of our patients, I think about 45% did receive that treatment. There were some nominal associations with some of the various forms of T-cell activation, but that probably has more to do with just how sick those patients were and not necessarily any direct effect of the drug itself.
Okay, great, thank you. The next question is, what types of pre-existing conditions do the adult patients have?
I'm actually really glad you asked that, because I forgot to mention it. So most of our adult cohort had significant comorbidities. I believe about 80% of them had some kind of cardiovascular pre-existing condition. I know about 40% also had diabetes, 45% were obese. And so our adult COVID-19 cohort had a lot of these underlying comorbidities. And this is really interesting because it was sort of in stark contrast to what we saw with the MIS-C patients, where those kids had absolutely no underlying conditions and were really robustly healthy. So that was sort of an interesting contrast in the two sort of severe forms of the disease that we're seeing.
Okay, thank you. And our next question is, do serum antibody levels correlate with disease severity?
Surprisingly, they do not at all.
Okay. And do the MIS-C patients have COVID antibodies?
Right. So I didn't show that data today, but yes, they do. So we've measured, again, antibody directed towards the spike protein in the SARS-CoV-2 virus. And all of our MIS-C kids are seropositive. And it's actually a defining feature of the disease.
Oh. Okay, we've got one more question. Does the CD4 T cell response also change over time in the MIS-C patients?
Right. So the MIS-C patients were characterized by both high CD8 and CD4 responses. And yes, the answer is yes. We also see a decrease over time from that first blood draw to the second blood draw in the CD8 and the CD4 responses.
All right. Well, thank you again, Josephine, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time. Goodbye.